Assalamualaikum and hello everyone. Welcome to our group presentation for SK22403 Instrumental Analysis Assignment. The title for our presentation is Phosphorus Analysis in River Water via UV Visible Spectrophotometry. Before we begin, let me introduce our group members.
Hello everyone, my name is Azrina Adwa binti Anip. My metric number is BS231101144. So I will explain the introduction of our experiment, which is a phosphorus analysis in river water. In this presentation, we will explain how phosphorus concentration in river water was determined using UV visible spectrophotometry. This method is commonly used in environmental analysis because it can detect very low concentrations of substances like phosphorus. Understanding phosphorus levels in rivers is important to assess water quality and ecosystem health. The main objective of this experiment is to measure the phosphorus concentration in river water samples. To do this, we first create a calibration curve using standard solutions of known concentrations. Then, we compare the observance of our unknown samples to the curve. We also analyze the results in triplicate to ensure accuracy and precision. And the importance of phosphorus monitoring is phosphorus is an essential nutrient for aquatic life. However, when it becomes too abundant, mainly due to agriculture runoff or wastewater, it can lead to eutrophication. This process causes excessive algal growth, which blocks sunlight and reduce oxygen in the water, and also can harming aquatic organisms. Therefore, monitoring phosphorus levels help prevent pollution and protects aquatic ecosystems. So, why use UV-vis spectrophotometry? Okay, UV-vis spectrophotometry is fast, affordable, and sensitive method for detecting phosphorus. It works by forming a blue color complex between phosphate and molybdate reagents. The spectrophotometer then measures how much light is observed by this complex at a specific wavelength. The more phosphorus in the sample, the more intense the color and the higher the observance.
Akida and I will be explaining how the river water sample were collected, prepared, and what reagents were used to detect phosphorus using UV spectrophotometry. First, water sample were collected from the mainstream of the river just below the surface using a acid wash polyethylene or glass bottles to prevent contamination. Before filling, each bottle was rinsed with river water to remove any residue. During sampling, we wore gloves and each bottle was labeled with the location, date, and time. Samples were kept in a cooler and transported to the lab, then stored at 4 degrees Celsius to preserve their condition before analysis. In the lab, the samples were filtered using a 0.45 micrometer membrane filter to remove solid particles. After filtration, a few drops of concentrated sulfuric acid were added to each sample. This step helps preserve the phosphate ions and stop biological activity. The preserved samples were stored in bottles at 4 degrees Celsius until they were analyzed. For phosphorus detection, we use the ascorbic acid method, which produces a blue color measurable by UV-vis. The reagent included ammonium molybdate, potassium antimonyl tartrate, sulfuric acid, and ascorbic acid. This reagent react with phosphate to form molybdenum blue, which is measured at 880 nanometer. For the standard solution with non-phosphorus concentration, it was between 2.5 to 10.0 milligram per liter were prepared and treated with the same reagents. Their absorbance was measured at 800 nanometer and a calibration curve was created to determine the phosphorus content in the river samples. Thank you.
Hello, I am Nurkiki Sharmila, metric number BS2311060, going to present the materials and methods part. To begin the UV via spectrophotometric analysis, we set up the instrument with a selected wavelength of 880 nanometers. This wavelength corresponds to the maximum absorbance of the blue-colored complex formed when phosphorus react with molybdate in the presence of ascorbic acid, a method commonly known as the molybdenum blue method. Before measuring samples, a blank solution is used to calibrate the instrument. The blank contains all the reagents except phosphorus, ensuring that the instrument zeroes out any background absorbance caused by the reagents alone. After blanking, we proceed with measuring a series of standard solutions with non-phosphorus concentrations. Their absorbance readings are plotted to generate a calibration curve, which shows a direct linear relationship between concentration and absorbance. This curve is later used to determine the phosphorus concentration in the unknown river water samples. Accurate measurements in UV via spectrophotometry require careful sample handling. Cuvettes, the small containers used to hold samples, must be clean and free of scratches. Before inserting them into the instrument, they should be wiped with lint-free tissues to remove any dust, fingerprints, or residue. It is important to handle cuvettes by their frosted sides, as touching the clear optical faces can interfere with the light path and affect absorbance readings. Also, before measurements, we must ensure that no air bubbles are trapped inside the cuvettes, as they scatter light and lead to inaccurate results. Lastly, to remain consistency, the same type of cuvette should be used for all measurements, standards, blanks, and sample alike. Thank you.
Next, we can create a clarification curve based on the data given. The table shows the standard concentration of phosphorus that we use to create the clarification curve. The concentration range from 2.50 to 10.00 milligrams per liter, and the corresponding absorbance values were measured using the UV visible spectrophotometer. These standard solutions are important because they help us establish a relationship between concentration and absorbance, which we will use to determine the unknown phosphorus concentration in the river water sample. Using the standard data, we plot a calibration curve with absorbance on the y-axis and concentration on the x-axis. From the graph, we obtain a linear regression equation y equals to 0.0255x minus 0.0067. The data showed a strong linear relationship, meaning that absorbance increases proportionally with phosphorus concentration. This regression equation is essential because it allows us to calculate the concentration of the unknown sample by inserting its absorbance value into the equation.
For the river water sample, we took 3 absorbance readings to ensure precision and consistency in the measurements. The absorbance values were 0.0086, 0.0091, and 0.0083. We calculate the average absorbance to be 0.00867. This average value will be used with the calibration equation to determine the phosphorus concentration in the river water. Using the regression equation, y equals to 0.0255x minus 0.0067 and the average sample absorbance of 0.00867, we rearrange the equation to solve for x, which represents the concentration. After substituting the absorbance value and calculating, we found that the phosphorus concentration in the sample is approximately 0.603 milligrams per liter. This value is slightly above the expected range of 0.1 to 0.5 milligrams per liter, which could indicate higher phosphorus level in the tested river sample. Lastly, quality assurance or QA focuses on preventing errors throughout the procedure. One of the main QA steps we took was using a well-established and validated method. We also ensured that the spectrophotometer was properly calibrated before use. This involved preparing a black solution containing only the reagents with no phosphorus to zero the instrument and eliminate background interference. Additionally, we used the clean cuvettes and followed a consistent procedure for preparing standards and sample to minimize variability and human error. Moving on to quality control or QC. This involved checking the accuracy of our result during and after the analysis. We began by preparing and analyzing 5 standard solutions to build a calibration curve. This helped confirm that the instrument response was linear across the concentration range. For the river water sample, we recorded 3 absorbance readings, which gave us values that were very close to each other. This shows good precision and repeatability. Finally, the absorbance value of our sample fell within the range of the calibration curve, which confirms that our result is valid and trustworthy.
In conclusion, UV visible spectrophotometry is a suitable method for measuring phosphorus in water. It is efficient, affordable, and accurate. Based on the results, the river water sample had a phosphorus concentration of around 0.1 milligrams per liter, which is within a safe and acceptable range. To summarize, we found that the phosphorus concentration in the river water sample was approximately 0.1 milligrams per liter. This value falls within the expected range of 0.1 to 0.5 milligrams per liter. This method shows precision based on the consistency of the triplicate measurement. The low phosphorus level in the sample suggests that the river is not significantly polluted. This is a good sign and indicates that the water is likely safe for aquatic organisms. Monitoring like this helps us detect any future increases before they cause serious environmental issues. Consistent monitoring of phosphorus is essential for environmental management. It helps detect early signs of pollution, prevent long-term damage, and support decision-making for water resource protection. Rigorous testing ensure the continued health of aquatic ecosystems and safe water quality.